Chapter 5, Tissue Organization, Part 1. So we are moving up from the chemical level through the cellular level, and we've now arrived at the tissue level, the tissues being the cells and the extracellular material around the cells. Cells start off as stem cells. These were totipotent stem cells back in the day when we were just a fertilized egg growing. Totipotent means that the kind of stem cell can become any other kind of stem cell possible. Then, as we developed, we became an embryoblast that was made of pluripotent stem cells. This means that they can become any cell except the placenta. So in our bodies, there might still be some pluripotent stem cells that can become any other stem cell, any other kind of cell except placenta. And then as we grow up, our stem cells become more specialized, become very specific kinds of cells. So we have multipotent stem cells that can become a few different kinds of cells, for instance, the ones that lead to our blood cells. And the unipotent stem cells are the ones that divide and only become one kind of cell, and that's it, which is pretty common now in our bodies. So the kinds of tissues that are present in our body include epithelial tissue, lining surfaces, nervous tissue for transmitting electric currents, muscle tissues for contraction, and connective tissues that do a lot of things. That's a pretty complex category. We'll start with epithelial tissue. Again, epithelial tissue lines every body surface, lines all of our body cavities. Anytime there's some sort of open space that is not filled with tissue, it is going to be lined with epithelial tissue. So inside of the hollow organs like the stomach, the intestines, the trachea, the lungs, and so on, the heart. Also, epithelial tissue makes up most of the glands of the body. And epithelial tissue is mostly cells. There is sheets and sheets of cells, and there's little to no extracellular matrix, except for the basement membrane. So here is a cartoon of a sheet of cells forming an epithelial tissue up next to an open space referred to as a lumen. These cells have polarity, basically meaning they are not the same on both sides. So their apical surface is exposed to the lumen, to the open space, while their basal surface is attaching to a structure called the basement membrane. And then underneath the basement membrane is connective tissue. So epithelial tissue is always connected by the basement membrane to underlying connective tissue. Epithelial tissue is avascular. This means it has no blood vessels within it. Epithelial tissue is highly innervated, so lots of nerve endings reach up to the epithelial tissue, allowing for sensations to be detected. And epithelial tissue is highly regenerative. Since they are exposed areas, they are frequently damaged, frequently losing cells, so lots and lots of cell division occurs to replace the loss of cells. All right, the main functions of epithelial tissue. First up is physical protection. They're providing an actual protection for external or internal surfaces. They protect from abrasions, from destruction, from dehydration, from chemical exposure. Just very, very important protective functions. Epithelial tissue is also selectively permeable. It allows some things to pass through the tissue, get deeper in the body, and prevents other things from passing through. Secretions, again, they form most of the glands, so they secrete, they produce things. That's what glands do. And then sensations, again, highly innervated, so they are a great place for receptors to detect various sensations such as touch, pressure, temperature, pain. As mentioned before, the layer of epithelial tissue that lies next to the connective tissue is lying on a structure called the basement membrane. So the basement membrane is the epithelial tissue structure that connects the cells to the underlying connective tissue. It is made up of collagen protein fibers, other proteins and carbohydrates. It's required, all epithelial tissue has it. Again, it provides physical support and anchoring site. The hemidesmosomes, formed by the lowest level of the epithelial cells, will attach to the basement membrane. So hemidesmosomes, again, the cell structure that attaches to the basement membrane. And finally, the basement membrane also can act as a barrier to help control the movement of large molecules into or through the epithelial tissue. Now, there are a variety of different kinds of epithelial tissue, and they are named almost always based on the layers of cells, how many layers of cells, and the cell shape. When we go by layers of cells, there are three names to remember. Simple epithelium has one layer of cells that are directly attached to the basement membrane. Then we have stratified. Stratified epithelial tissue has two or more layers of cells, with only the lowest layer, the deepest layer, attached to the basement membrane. And then pseudostratified is the third kind. It's sort of single layer of cells, but their nuclei aren't lined up, and it's kind of confusing. It looks like they could be multiple layers, so pseudo for fake layers. And then you also can define epithelial tissue by cell shape. So it could be squamous, which is a nice flat cell, cuboidal, a cell that is as tall as it is wide, and columnar, a cell that is much taller than it is wide. And when you define epithelial tissue by cell shape, it's always the cells near the surface, near that lumen, that open space. So for instance, here is some epithelial tissue. What would be the name of this epithelial tissue? Well, go through the columns. Simple, yes, simple, one layer. Squamous, yes, squamous because it is flat. So simple squamous, flat, one layer of cells. Simple squamous epithelium is able to provide the thinnest possible barrier. So it allows for rapid movement of molecules through the simple squamous epithelium. It is found in lining the air sacs of the lungs, allowing oxygen and carbon dioxide to pass. It lines blood vessels and lymph vessels, and this also forms part of the serous membranes that line serous cavities. You will see terms occasionally endothelium and mesothelium. These are exactly the same as epithelium. They just come from a different cell lineage, but they're basically identical to epithelium. So just so you're aware of that. All right, simple cuboidal epithelium. One row of cells that are about the same width as they are high in height, attached to the basement membrane. They are ideal for forming structures of small ducts and glands. They're able to absorb fluids across their apical surface, and they can also secrete things. They're found in the walls of kidney tubules. So down here, we're forming walls of kidney tubules. They can form the secretory regions of most glands and also smaller ducts and exocrine glands. And they also cover the surface of the ovary. Simple columnar epithelium also has secretory and absorption functions. There's two big kinds, non-ciliated. Non-ciliated simple columnar epithelium lacks cilia. Sometimes they do have microvilli, though, which can look like a faint little fuzz on top of the cells. So many non-ciliated simple columnar epithelium have microvilli. And often they also have unicellular glands called goblet cells. And these goblet cells produce mucin, which when mixed with water becomes mucus. So goblet cells produce mucus. And examples of locations, it lines the digestive tract, the digestive tract from the stomach to the anal canal. Then we have ciliated simple columnar epithelium, has those long hair-like structures called cilia. The job of the cilia is to move materials outside of the cell. Often that is mucus that's being produced by goblet cells. You can find ciliated simple columnar epithelium in the bronchioles found in the respiratory system, also lining the uterine tubes. Pseudostratified columnar epithelium also comes in ciliated and non-ciliated forms. Again, it looks complicated, looks like it might have multiple layers, but actually it doesn't. You can find some cells that go all the way from the bottom to the top. Pseudostratified columnar epithelium has the nuclei all scattered about. It's kind of confusing. Ciliated version also has goblet cells that produce mucus. Mucus is moved by the cilia, found in the large passageways of the respiratory system. Non-ciliated pseudostratified columnar epithelium doesn't have goblet cells and occurs primarily in the male urethra and epididymis. Stratified squamous epithelium has many layers. It's stratified, and if you go to the surface of the tissue, that's where the cells are very flat. So again, look at the cells near the open space to determine the shape of the cells. Simple, stratified squamous epithelium is big on protection. It protects against abrasions and frictions. And its basal layer, those cells, that's where all the cell division occurs, and those cells produce a lot. You have the non-keratinized form. These are found in moisture areas of the body, lining the oral cavity, parts of the pharynx, the esophagus, the vagina, the anus. Again, no keratin protein, but they are protective. They lose the surface cells when friction occurs. And they are alive all the way throughout. You can tell that because you can see the nucleuses all throughout. Non-keratinized stratified squamous epithelium has superficial cells that are dead. So no nuclei here. These cells are dead. Instead, they are filled with keratin protein, making it very tough and protective layers for the skin, the epidermis of the skin. Stratified cuboidal epithelium, again, 2 or more layers, usually 2. They form walls of ducts for many excretory glands, and they are able to form strengthened walls that can be larger, have more space to allow materials to pass through, and they're also found in some sections of the male urethra. Stratified columnar epithelium, so again, 2 or more layers, usually just 2. The more superficial layer is columnar in shape. Again, protection and secretion functions, found in the largest of ducts in the salivary glands, and also in parts of the male urethra. And then, surprise, we have one more. It's transitional epithelium. Transitional epithelium lines the urinary tract. In its relaxed state, the cells are sort of spherical in shape, but as pressure builds in the, say, urinary bladder, as urine fills the bladder, the cells will stretch and look squamous, so transitioning from the relaxed, rounded state to the distended, more flattened state. And this accommodates how the bladder needs to be able to stretch as it fills with urine. All right, so simple epithelia, their jobs is primarily diffusion, absorption, secretion. They allow things to pass through or produce things. Stratified epithelia, their functions are more structural in nature or protective, so protecting against friction, protecting against chemicals, or just providing a structure, such as a large duct. Glands, as I mentioned, glands are made of epithelial tissue. They can be single cells or multicellular structures. They secrete stuff. They can secrete mucin to make mucus. They can secrete electrolytes, hormones, enzymes, lots of possibilities. Endocrine glands lack ducts. They do not send things to a surface. Instead, they secrete hormones that go into the interstitial fluid around the gland or directly into the bloodstream. Hormones go out, find cells with receptors for the hormones, cause things to happen. Exocrine glands, on the other hand, are invaginations of the epithelium, and they include a duct that's going to go to a surface. So they secrete materials that go to a surface, either inside the body or on our surface of the skin. And they can produce things like sweat, gastric juices, and so on. Glands can be kind of complicated in structure or simple in structure. Simple glands, just sort of a tube, maybe a slightly branched tube, could be coiled, rounded. Compound glands are more complicated with multiple branches that then branch even more. That's all we're going to worry about. They can be simple, they can be complicated. How do they secrete things? Well, you can do it by American secrete glands produced by American secretion. This is exocytosis. They produce stuff inside the cell and then release it via exocytosis, vesicles releasing their contents into the space. American glands. Then we have apocrine glands. They are able to produce the materials in the cells that then form on the apical surface. And then when enough material is there, they bud off. So you have little globules of cells filled with the extracellular secretions. So that's apocrine glands. And then holocrine glands. Here, the cells near the space inside the gland fill the secretion. And then when it's full enough, those cells will disintegrate, becoming cell bits that then get released. So holocrine glands, the entire cell, whole cell, is being broken down and released as the secretion. And that is it for this lecture.